Okay, good day everyone. Nice class size today. I like it. Uh, Picking up what we have left previous week, we are going to continue on looking at the internal structure of eukaryotic cell. Now it's time to take a look at the cytoskeleton. Cyto always means it's cells. Okay, so it means literally cells skeleton. Assuming we all know that what skeletons are internally, and the functions of skeletons are what? Supporting. Without any skeleton inside the bones, our shape not have been possible. So that's exactly what this so-called cytoskeleton would provide us the function. Probably one of the most obvious function of this cytoskeleton is to support and then maintaining the shape of any particular cells. So one obvious thing about this cytoskeleton is this is again a unique feature of only eukaryotic cells. So plants and fungi and animals all have these cytoskeletons, but those prokaryotes like bacteria do not have such. May I call this as sophisticated? Maybe, yeah. These are sophisticated structures. And another thing is, excuse me. Another thing is, this is not something that, unlike what we have looked in the previous week's lecture, this is not not an example of the typical eukaryotes. What the organelles. The cytoskeleton is not an example of organelles. It is not. Okay. They are simply some complex proteins. They are just proteins, but not the organelles. The difference between proteins and organelles are obvious in that organelles are basically membrane-bound organelles. Any organelles inside of eukaryotic cells are enclosed with their own territorial membranes, whereas these cytoskeletons are devoid of any of such membranes. So they are strictly just the proteins only. Okay. So anyway, let's take a look at see if let's see if that works. If not, so once again, no audio. So never mind. So only this just impressive looking. Hey, what is the audio? Whatever. Okay, so one thing is this is the network of protein filaments. The wording filaments. So it's a long thread-like structure. In contrast to something, there are two contrasting, structurally speaking, shapes of a protein you can imagine in our world of proteins. One would be some roundish shape of protein, roughly speaking, in general. This type of protein we would call a globular, ball-like globular proteins. On the other hand, long, like this filament-like long thread-like protein, we would call fiber-like fiber. So fibrillose. So these are generic These are two opposite generic terms describing two different alternative shapes of proteins. So anything, whenever you were encountered with the description of globular proteins, etc., blah, 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 and oh, okay, so those protein shapes are roundish. Okay. So there are so many examples of such globular proteins, including hemoglobin. Guess why hemoglobin named hemoglobin? Globin. Always IN. Anything ends with IN, you just guess almost 90 out of 100 or 99% out of 100, you can guess correctly that would be the name of a protein. It will end in IN, globin. So hemoglobin means some heme. Heme is something that hemoglobin contains in any particular structure, just like that. So anyway, but on the other hand, these proteins are not round, but very long, and that is something that needed for their respective functions, all these cytoskeletons. So as I read, it maintains the cell shapes and provide other structural supports, just like our skeleton. And another thing is these three important functions we can summarize So this cytoskeleton, in addition to provide and function as the support to maintain shape, it also function as 1R, 2R, probably 3R. So the first R is road. Maybe should I put plural? Roads. The spider web-like roads are actually aligned in each of our cells, and all of which are made by the lining of this cytoskeleton. So it functions as a road. So provides passages for transport of the cellular components. That's what it is. So the previous week we looked at that exocytosis, all those materials in the form of transport vesicle. Still remember the transport vesicle? Those transport vesicles are actually walking along the lines of all the cytoskeleton. It's not simply floating in the whole volume, just like a water balloon. No, it is not. They are not. They are exactly walking the path, exact particular paths aligned by the cytoskeleton. So in that way, it functions as a road for all these cellular components transport passages. And it also controls the cell division when they are about to divide the cell division. The chromosome have to separate it. Okay, so and also two cells, especially in the case of an animal cell, the two cells must be squeezed to separate them physically. So in that way, probably we can describe their function as a what? What do you think? Rope. So pulling, hey, grab chromosome and then pull one direction so that each chromosome gets separated. So this cytoskeleton works like rope-like function. So it is like a Swiss Army knife. It has so many different functions. And third, directs the cell's movement. Is this another R? Actually, it's like a propeller-like. It's not a road. So it's my bad. It's my mistake. How can I just describe this function as starting with the R? Anybody? It's a good idea. It provides some movement function. Maybe rotor. Why not? Rotor, like moving part. So it's outside of any each cell, it provides any some mobility. Okay. So that this cell having this external cytoskeleton can swim around, or it can provide some waving functions. So all those moving. So whatever these are, start with R or something else, doesn't really matter as long as you understand the basic three additional functions. Okay. Three additional functions of the cytoskeleton. In the previous slide, we just looked at the typical, stereotypical function of supporting, okay, shape, maintaining the shape as a like skeleton. When you imagine skeleton, you can easily imagine their function to be maintaining the shape and the supporting role. But that's not it. In the case of a cytoskeleton, that's just one thing, but extra gravies are just like this. And this is not even the end. There is some more. And this muscle flexing, contraction, is also a job of a particular type of a cytoskeleton. So animal, plants, unfortunately, plants do not have this muscle contraction. So this is another privilege of animal cells only, especially among those animals, only the multicellular animals privileged. But anyway, all this, the reason why we can move around and then do this all kind of planned activity, like sipping my beloved coffee during the lecture, which is the best part of my lecture for my individual personal sake. But these are all the job provided from a particular type of cytoskeleton. And dynamic assembly and disassembly, forget it, because this is too much. This is very important. Dynamic assembly and disassembly is very important feature of cytoskeleton, a particular type of cytoskeleton. But if this were cell biology course, of course, definitely I would have emphasized the importance. But fortunately or unfortunately, this is not a cell biology course, so I will, okay. I will not put you responsible for knowing this. Forget it. So there are three types. So cytoskeleton, all these three different types of a cytoskeleton, one common denominator is they are all long, fibrous, filament-like proteins. They are very long. It's not globular, round. Another common characteristics shared by these three types, all three types of cytoskeletons are also, these are polymers. Once again, do you still remember the definition of the polymers? All those polymers must be assembled starting from what? Individual unit we call monomers. Okay. So each different types of cytoskeleton has its own particular monomeric component, the form. So once again, if this class was a cell biology course, of course, it's very important for you to remember and distinguish, being able to distinguish what are each different type of monomeric form and then how they assemble to form these. But luckily, this is only introductory 교양 life science course. So you don't know, who gives a damn about those little miscellaneous trivia? So forget it. Or just the only important thing is there are three different kinds, and each of which has their own each version of a particular component from which these long polymeric forms are assembled. That's enough. That's all you need. Okay? But anyway, so let's go. So in the case of actin, this reddish one particular using a particular, if you understand what the fluorescence is, we are getting there in this chapter later on. But anyway, this is a particular form of a light source called fluorescence. So we can stain a particular component with a particular color of fluorescence light. So in this, all this red color indicates the presence of this particular type of the cytoskeleton called actin. Actin, three different forms, one of which is actin, the other one is a microtubule, another one is an intermediate filament, with three different names. Even you don't really need to know three different names, who cares? But there are three different forms of cytoskeletons that are the only things important for you to carry and as a take-home message. But anyway, so this is a typical animal cell, our own cell. And if you want to visualize, I want to know if you are curious about, okay, where exactly whatever this actin cytoskeleton are present, and it's all over the place. Surprise, surprise, surprise. It's everywhere. Oh my gosh. Okay. So cell itself is a so crowded structure. Okay. On top of many, many different types of organelles, you now have literally, it is a really dense spider web of cytoskeleton, and this is only one of three different forms of cytoskeleton protein. So does that mean, okay, this particular cell only whatever it is, this actin microfilament is only present? No, it's not, of course not. The same cell probably you would have also observed many other. This is another type called the intermediate filament, another type of cytoskeleton. Once again, it's everywhere. Oh my gosh. How would they distinguish all these different lines of cytoskeleton? It's like mind-boggling complexity. And in the case of a microtubule, this is the biggest one amongst those three. That's why the biggest, and it's formed the hollow tube-like, the big structure, if you want to know. Well, but anyway, these so-called microtubules are also everywhere. So that's why, and that's how they provide so many different variety of functions, including like a Rhodes function and muscle flexing, like contraction function, and these rope-like pulling functions and squeezing function. Oh my God. So this kind of summarize really nicely, sum up, trying not to confuse by giving too much complexity. Simply, for example, roads. One such form of cytoskeleton provides this big highway-like road on which each different transport vesicles move from. So this is in the course of so-called exocytosis, starting from this ER, rough ER, Golgi, and they are moving, going to the outside. Oh, nice cartoon. This is one particular figure. I took it from very famous cell biology textbook. Okay, so and so if you wondered how this is a transport vesicle. How this transport vesicle can correctly move along this road like cytoskeleton? Because if you zoom in what's going on, like this, actually at the bottom of this each transport vesicle lies a particular protein working only on the surface of this cytoskeleton function. We call that as a motor protein. And this motor protein, it's spending energy. What kind of energy can you guess? What else? It's ATP. ATP is everywhere. Spending ATP, like using ATP hydrolysis, ATP breaking down, so they can walk by one step, one ATP, one step, one ATP, one step. That's how they can do this. Yes. So all those are the functions of different types of motor proteins. And all these motor proteins are on each rod-like cytoskeleton. And that's the reason why any cargo that we need to ship can move along the cytoskeletal road. Okay? And why are you still in this? Okay, now you are talking. So different cytoskeletal proteins are aligned on different location so that all together they can coordinate and then cooperate so that each different part of any given cell can have their needed this transporting passages is another feature. And this is a part of the neuron cell. I don't know. So one of the reasons why we can transmit our nerve signal from one part to the long distant location is because of this structure. If you knew typical structure of a nerve cell, I can draw roughly. It looks like this. This is very atypical shape of a cell for even an animal cell. And a typical animal cell would look like this, right? But this nerve cell looked like very distinct morphology, so to speak. And this is the direction, always. This is the head area. This is the direction always from head to the tail, head to foot, direction of a nerve impulse. And so this is a long extension of a cell, okay, surface itself, along which this, whatever it is, a signal must travel from head area to the tail area. And this will, hey, can you, never mind. And another, the second nerve cell is being connected so that this nerve signal can continue on to transmit. So that's how we do. But this is a part of this particular structure of a nerve cell. And what we can see inside of this structure is a whole bunch of the wire-like, electric wire-like cytoskeleton. And that is the reason why this signal can travel thanks to this cytoskeleton. So if that's too much, why do I have to know all these unnecessary details? Okay, fine then. The only thing is, okay, this is another such function. So even cytoskeleton is needed for our nerve signal transmission. Would that be enough? That's it. Okay. And this is when these two cells is about to divide. So obviously, when cells, animal cell, plant cell is a little bit different story because plant cell has an additional rigid protective layer outside of the cell membrane. We call the cell wall. So they have a little different technical problem. But anyway, animal cell, any typical animal cell, if this cell is going to divide, and this would like first, this would be the plane of this division equator, so to speak, and once again, this cell would pinch. Okay, so at this, gradually, this cell would try to separate into two. So this particular equator line would pinch in, eventually further squeeze down to until get separated into two independent cells. That's how you go through typical animal cell division. So that's where this rope-like cytoskeletal function is in action. So wrap this area, this is a cytoskeleton, particular cytoskeleton. It happens to be an actin, but who knows? Who cares? So this cytoskeleton will strap and then contract just like the same way they contract our muscle fibers. They will contract so that being squeezed in further and further to get separated. So that's one rope-like function. Another, in the meantime, at the same time, this outside cell shapes, the cell membrane being separated is one thing, which is very important, but that's not all. Probably perhaps more important task during the cell division would be you have to divide. You have to divide each chromosome. These are the chromosomes. So that they distribute their whatever they have, the chromosome, into two equal halves, so that each dividing cell inherit exact same number of same kind of chromosome. How? How are you going to divide, separate these two chromosomes? Here, this blue, sky blue lines represent cytoskeleton, another type of cytoskeleton. Each attach individual chromosome and then pull. Okay, come on, this side. So, due to that action, each chromosome will migrate to this direction as well as to opposite direction. That's how you separate your genetics whole information. So these two routes, two separate important route of cell divisions are all managed by the function of a cytoskeleton too. Okay? And I'm not even going to touch. And last week also, we briefly kind of discussed that all particular type of white blood cell in our body can eat bacteria, right? This phagocytosis. And actually that kind of cell eating activity was originated from this guy, one of my favorite animal, amoeba. Amoeba is actually very renowned or notorious for such cell eating activity. And those characteristics of amoeba was transmitted into some particular type of white blood cell in our multicellular organism like ourselves. So each this little projection on the surface of amoeba can be extended, boom, and grab any object. So in the case of amoeba, that would be the target would be their food. In the case of our white blood cell, the target would be any invading cell, bacteria cell, or virus. But anyway, the same thing. So this extracting quickly and then retracting so quickly so that they can grab and then internalize this thing is all how they do that. Rapidly cytoskeleton protein inside are synthesized. Can you imagine? Rapidly you just build up long thread of cytoskeleton so that thanks to that this particular surface can be reached out to grab. And then now what? You rapidly degrade this synthesized one, then it will be retracted. That's how you do it. So that's another job of cytoskeleton protein. Whatever. Okay, so that's probably the end of the discussion of cytoskeleton. Yeah, although we can talk whole night about different kinds and different mechanisms of cytoskeleton, but obviously that's not a productive thing. So we can just, it's me or, so let's take a look at ribosome now. Why do I have to throw out this unnecessary information? What's ribosome? Let's forget about all this. What's ribosome? What are ribosomes? Ladies? Protein synthesis machinery. Great, thank you. Right, exactly. So this is relatively big, bulky part. Do you know, or can you tell us what's the ribosome made of? Proteins. Yeah. Anything else? So it's a mixture of RNAs and proteins. Whole bunch of several, something like 20, 30 different kinds of proteins mixed with several, 3 or 4 different RNAs. Then they mix together, and voila, this is ribosome. That's why in the ribosome, one particular type of RNA acts as a ribozyme, which is the RNA acting as an enzyme, kind of. So anyway, that's it. But here, prokaryotic, eukaryotic, 70, 80s, whatever, forget it. This whole thing, whole information is, the essence is prokaryotic ribosome, although when you look at from a distance, both prokaryotic ribosome and eukaryotic ribosome, they look so much alike. And they are same ribosome. And they are also, both prokaryotic and eukaryotic ribosomes are all made of the combination of several proteins plus several RNAs. Yeah. But even that, they are different. Prokaryotic and eukaryotic ribosomes are different. That's all we need. How different? Obviously, first, eukaryotic ribosomes are bigger than prokaryotic ones. And so fine detailed, the structures are also different. That's all we need. So they are different. Okay. So this is a typical cartoon of a ribosome. Fine. So in other words, There are two parts. We called large subunit and small subunit put together one functional entity of ribosome. But even that one is not even necessary for you to remember. So what? So this is another description of how prokaryote and eukaryote ribosomes are different. Don't don't be fooled by any detail. Just yeah, they are different. That's just enough, right? Now, because of that, this ribosome is a very good target for making, developing any antibiotics. That's the purpose of my intention. So where are we getting at? Yeah, I want to talk about this, antibiotics and how effectively, efficiently, how can we kill any those nasty, ugly, the bacteria that we hate. Uh, wow, what is this? This is one summary of major only major. There are so many other different types of antibiotics. Here, how many of these names of antibiotics, all these names of antibiotics are in pink? Uh, how many of these antibiotics, different antibiotics, can you recognize? Oh, I have heard once before this. Probably at least one. Here. Maybe this one? What do you think? This? How do you pronounce this one? Penicillin. Have you heard of a penicillin before? Yeah, probably, because that's very important antibiotics. What's antibiotics? What's antibiotics? Yeah, 항생제, of course. Yeah. What's that? A drug. It's drug, which is good for our health. Is it? Wow. The antibiotics, by definition, is that drug designed to kill or inhibit the activity of only the prokaryotes, only the bacteria, leaving such similar activity or metabolism of eukaryotes. So eukaryotes activity and then viability should not be affected by antibiotics. There shouldn't be anything like antibiotics, and they can influence both eukaryote and prokaryotes, by definition, at least theoretically. Because by definition, antibiotics are only for killing or inhibiting the bacteria, prokaryotes only. So that's why I brought up that the ribosome can be a very good target for developing antibiotics. Because what did I say about ribosome? Prokaryote ribosome is different from eukaryote ribosome. So if we can develop any drug that can only affecting the functionality of a prokaryotic ribosome, leaving this eukaryotic version of ribosome intact, then that can be a very good, efficient antibiotics. So here, the reason why I brought up this penicillin is that is what? Historically speaking, that is the first antibiotics ever developed. Actually, it was accidentally discovered. It's not made from, but it was discovered from because that penicillin antibiotics happened to be made by a specific type of fungus. The fungus will be eukaryote, and the fungus will gladly kill any bacteria. That's how they somehow, through evolution, they have acquired this particular strong weapon to kill any bacteria. And then people discovered this penicillin, and it was such a blessing. But so imagine the era of pre-antibiotics, when the antibiotics were not available, you have a even minor injury, especially during battles. Okay, a lot of people, a lot of wounded soldiers, they have to go through this amputation because such a small injury, but there is no way you can control this infection contamination, and then these bacteria will just take over all entire areas. So the only thing you can save your life is just cut it off. But once you have this strong ammunition of antibiotics, you don't really worry about. So you can so efficiently kill this uh, infecting bacteria. And by the way, penicillin, on this cartoon, what do you think? Penicillin, their function, the activity of penicillin is achieved through what? In biology, always this particular type of arrow, black arrow, indicate what? Inhibition, blocking. So if you see this arrow, particularly going to this, what is this? Cell wall. Cell wall is what? Do we have cell wall? We don't, luckily. Only bacteria have cell wall, chemically, which is very unique, distinct from other cell wall barrier bearing the organisms like plants and fungi. So this penicillin happens to be inhibit cell wall synthesis. You don't have to know this, but anyway, just in case you're curious, what's the function of how does this penicillin work to kill the bacteria? That's how it does. So penicillin and the same type of the antibiotics or the vancomycin, both of them work by inhibiting bacterial cell wall synthesis. If any bacteria cannot make their cell wall, they cannot grow, so they are dead meat. Okay, this is a very effective way of containing the bacteria. And once again, so by the same token, you can figure out the different antibiotics, how they work. For example, this, whatever, quinolone, what does it look like? DNA. Bacterial DNA. One bacterial DNA is being converted into two. What is it? One becoming two? What's the procedure, particular name of this procedure? One DNA become two DNA. What would you call? Come on. That's DNA replication. So any dividing cell must first take care of this DNA replication, right? And what does it mean? This, whatever it is, quinolone can block DNA replication? How? How would any drug can stop DNA replication? It's all in the enzyme. There's got to be a particular enzyme responsible for making this DNA replicated. We do have the particular such name. We call that the DNA polymerase, but who cares? But this bacterial DNA polymerase is the target of this quinolone. So what's the message of this? So quinolone, whatever it is, I keep saying this. This particular antibiotics, the reason it can be a very effective antibiotics is because it can only affect bacterial enzyme, DNA polymerase. But our own version of DNA polymerase is not going to be affected by this one. And that's why we can safely rely on these antibiotics. Should I go on and on and on? No, it is not the reason. Any particular trivial information is not at all important. We do not have to know penicillin is the one that inhibiting the cell wall synthesis and quinolone is the one inhibiting bacterial DNA polymerase. No, that is not for you. Okay. The whole entire message is what? This is particular area, potential area, that area of metabolism, prokaryotic specific metabolism, and because of that, there are several antibiotics already available, each of which specifically targeting on this prokaryotic specific metabolism. That's the whole thing to understand. So that many different metabolisms are. And these, basically all these metabolisms are also shared by us. So we do also have the same metabolism, like this ribosome, this is a ribosome, and ribosome is doing this protein synthesis. We do also have a ribosome, and then we do also need to make proteins. But still, all these different antibiotics can target specifically for this is what? Their mechanism, especially bacterial ribosomes, are different from eukaryotic ribosome, so we can easily forget about any potential danger. So now, probably we are coming back to consider some specific problem of this antibiotics related one later in this chapter. But for now, let's focus on this nucleus. How do we have a nucleus, some specific area nucleus? Maybe it's a very silly question. Like, what is nucleus? Is it organelle? Another organelle? Yes. So in other words, nucleus is also what? A membrane-bound structure. So it is a membrane with particular area enclosed the membrane. Now we call that specific area as a nucleus. And what's in the nucleus? Inside of the nucleus, we do have our precious genetic material, DNA. DNA is there. That's all. So nucleus, when we try to think nucleus, okay, we do have some specific area, specifically designated as a storage place for our genetic material, inside of which our genes are there, and their particular activity is always going on, which is very important because we need to store them and then take care of them very safely because that's very important. Our genes, that's it. So that territory, boundary, is a nucleus. Another thing that we need to probably consider is then probably some of you at least might wondered, okay, that nucleus is one particular territory inside of which is very important activity is always going on. As a result of that activity, we do have some specific products. That specific product must come out of nucleus to the cytoplasm, cytosol. And also, in order to support that activity from the outside, which is the cytoplasm, cytosol, we need to provide some material, raw material. How do we do that? How do we coordinate such material traffic between inside of nucleus and outside of nucleus? Probably that's another thing that you might want to actually learn. Okay. Is there any specific way? Do we have, because this is one big difference. If, let's say this is a cell, and this is a nucleus, there is a material, very shiny star material outside of a cell. It wants to go in. Okay. So this is one particular example of a traffic. How usually what do you think? This star material would be handled if this star wants to come in to the cell. How do we take care of it? Especially if this star is hydrophilic. Star is polar. Then we do have a certain specific rule. What is this? Receptor or transporter? Right, probably transporter that can specifically recognize this star, and then, hey, you can come in through this particular passage. That's how we do it. So in other words, on the membrane, we take care of all this material transport very specifically, one-on-one based. One-on-one. Individual basis, I pick you, I recognize you, then I will allow you to come in or to come out type of this specific relationship using. That's why we have so many different proteins as we have seen previously, right? So my question is, do we have same or at least similar principle working on this surface of this nucleus? Same. So if this star, once again, hey, I want to take a tour of the inside of the nucleus, I want to go in, and does this star have to go through exactly the same routine as it experienced on the surface of the cell membrane or not, is my question. Probably you don't know, right? But just take a guess. What do you think? Same or different? Different. Nucleus is very generous. Cell membrane is very picky. But nucleus is, well, easygoing. Hey, what the heck? Everybody is welcome. You can come in. But not everybody, actually. So nucleus has a small hole. On the surface of the nucleus, we call it nuclear envelope. This surface we call as nuclear envelope, like a nuclear membrane. The surface of nuclear envelope contains so many small holes. We call nuclear pore, through which any decent medium-sized, small to medium-sized material can easily go in and come out. So that's why I can say nucleus is very easygoing, generous. That's one big difference. Okay, that's the difference and how they manage the traffic of material between outside and inside of the nucleus. And another important information regarding this nucleus is what's inside. We all know that inside of the nucleus we do have our gene. In, in eukaryotes, this our gene are organized in terms of its physical structure. They are organized in what? The name we call chromosome, isn't it? Chromosome is the structure which contains the DNA, right? DNA. Why do we distinguish like a chromosome from DNA? Isn't chromosome the same as DNA? Tell me. Is DNA is exactly equal to chromosome, or is there any something else is there? Is my question. DNA equals chromosome. DNA is not equal to chromosome. Which one? DNA is not equal to chromosome. Chromosome contains DNA, fine, right. But chromosome also have additional one, which are protein. Chromosome is made of half of the chromosome, the entire structure of a chromosome, half of them is actually contributed from proteins, and half of them probably what we can say is like DNA. So this chromosome is a structure containing the DNA, and the importance of which is because DNA serves as our genes, but there is something more, which is the proteins are also embedded in. And so this is what DNA is wrapped around. This is the name of that generic, most common chromosomal protein, the histone. Can anyone guess the chemical property of this histone proteins? Like, for example, histone, as a histone protein, what kind of amino acids does histone protein have very abundant in terms of a chemical property? Like positively charged or hydrophobic or negatively charged? I will give you three choices. Histone protein would contain this particular amino acid very abundantly. This is a statement. And example, 1, positively charged amino acid. 2, hydrophobic amino acid. 3, negatively charged amino acid. You pick one out of 3. You have a very high chance of being correct simply by guessing. What do you think? Positively? How did you know? Okay, you why don't you stand and go out of you go home. You don't need to remain in the classroom. Thank you. Right. So, yes, DNA is highly negatively charged. Remember, DNA is extremely negatively charged. Okay? So, anything trying to associate intimately with DNA, you better be oppositely charged one. So that's why this histone protein is highly positively charged, thanks to the proportion of positively charged amino acid is so high. Okay, anyway. And another thing is, even if you are not a science major, or even if you are not especially the life science major, you better be able to distinguish. It will be really good for you to be able to distinguish the difference between chromatin and chromosome, because by doing so, you can understand the organization of this our genetic material as a chromosome. So chromosome is a generic term. We just think of a chromosome, just a chromosome. But so if I ask you to draw a typical chromosome, can you draw a typical chromosome structure? Then probably it would be like this. This is a typical, or even some of you may even draw like this. Either this or that. I will draw for now this version of thing. So this is a typical shape of chromosome, but this is only eukaryotic chromosome. Bacterial, prokaryotic chromosomes are not like that. We have already seen that. Prokaryotic chromosomes are what? Circular. But eukaryotic chromosomes are linear. Okay, that's the big difference, and then that's a really important difference. But anyway, forget about this circular versus linear things. But my point that I like to make here is this typical shape of chromosome, eukaryotic chromosome, is actually, you would be very lucky if you actually really want to observe any given cell under the microscope and try to observe, okay, I want to find this chromosome structure, then if you succeeded in finding this the chromosome structure that you want to see, you are very lucky, because this doesn't exist very frequently. It is very, very short period of time chromosome exists in this way. Most of the majority of the rest of the period, chromosome, this is typical chromosome structure, but actually the chromosome exists as this chromatin structure, which is different. This is a chromatin versus a chromosome. Can you understand? So this is the same thread, but we can wrap and organize this thread tightly wrapped, or you can let it go loosely unorganized, like pulled out. So this is the difference. This is chromatin, and this would be chromosome. Depending on different occasion, different particular period of the timing in a cell, chromosome exists either in this chromosome, highly condensed. Chromosome is a highly condensed form like this, and chromatin is uncondensed, loose form. On which occasion would you rather want to tightly wrap your chromosome? At what particular occasion? Can you guess? You have like entire different lifetime of a cell as a single cell, which we call as a cell cycle. Cell cycle, by definition, is the entire lifetime period of any given cell. So once one cell become two cell as a result of a cell division, the cell cycle of this particular cell will end, and then each of these two new cells will start another new round of their version of cell cycle, whatever. But anyway, that's not important. But during the entire cell cycle, at which particular period or point would you rather want your chromosome exists in this tightly packed, condensed one, whereas this, hey, easy going. Okay, life isn't like a life is an easy one. Okay? Relax type of loosely uncondensed one. Any particular point or occasion you can think of that you better, oh, I better wrap my chromosome tightly condensed like this. Anybody? Cell division? Why? Why do you think that cell division is like? Cell division is just like when this cell is divided into two, it's like in our everyday life, it's very similar to our moving day. You want to move, then you better pack all your stuff. Before you leave, when you live your normal life, you don't really take care of cleaning your room or whatever. Everything is just scattered around. But hey, this is the moving time. Then you gather everything and then pack into tightly spaced one so that you can easily pick so as not to lose any precious one. Exactly, yeah. So just before the cell division, this is the only occasion, point that your whole chromosome become real chromosome, like highly condensed one. In other words, for the majority of the rest of the cell cycle, your chromosome will exist in this, which, what did I call this status? Chromatin. Okay, that's it. If you can understand this, that's probably very important the information that we need to cover. And this chromatin, this is maybe a little bit of a, so how many in terms of a chromosome, how many different chromosomes do we have? Kinds of chromosome. How many different kinds of chromosome do we have? 20, 24, is it? Or 23? We have one more kind of chromosome? Oh, you mean the X and Y is a different one. Great. Yeah, gotcha. Okay. So anyway, but typically we say like we have a 23 different kinds of chromosome. Okay. You know, uh, especially in your case, only 23 different kinds of chromosomes. I have 24 different kinds of chromosomes. Yeah, yeah, but not you. But so such different chromosome existing in the form of chromatin, obviously, they have. So you would think like, yeah, these chromosomes are all mingled together, like they are chromatin anyway, loosely organized. So probably that's what you would imagine. Most of the time, your chromatin would exist like this, but that turns out not to be the case. Even during this period of unorganized one, as a chromatin, they have strictly have their own unique territories. Just like those Americans and Europeans, they have their such a very strict sense of territory. So in one town, it's very surprising. One time, just one block away, this area is so safe. Everything is so happy, seemingly at least. But you just one step over this block, and this area is just like full of criminals and so dangerous. So surprised. I could not even understand how these things are all maintained. That's why their culture, they have such a strict territory concept, unlike us. And maybe chromosomes, they learned this from the chromatins. So this, uh, some scientists developed some different color of dyes. Each different color dye can stain different type of chromatin. So by doing so this is a nucleus, human nucleus. And what are these different colors? when you apply such different dyes, each you could find very distinct, different colored area, which represent chromatin territories, just like different blocks of those towns. Wow. So that's what we now realize that, okay, chromatins, even as a chromatin stage, they maintain their certain very distinct territories, chromatin territories. Okay, big deal. Good. So now we know, but that is not the reason why we need to go through this. More important thing is, what if someone walks in and then mess with this, so that what chromosome or chromatin territories, why do we need this? So hey, disturb all this, and from now on there are no such chromatin territory in my territory. And then they just mess everything around. And what would happen is my question. Would that be something, okay, fine. We do have somehow traditionally, we maintain these chromatin territories, but if someone mess this thing around so that we disrupt this so-called chromatin territory, nothing happens? Or is it something very serious, so that if someone mess around this chromatin territory, then this cell would die or anything? That much is serious problem? Which one would be the case? Obviously, of course, the latter would be the case. That's why I am bringing up this. This chromatin territory happens to be very, very important, critical in cells' viability, survivability. Turns out that. This is the outcome of such disruption of a chromatin territory. Progeria is, oh, okay, I didn't put. Progeria is a genetic disease that causes premature aging. So this young lady, probably at her teens, probably 12 or 15, or I don't know exactly, I don't remember exactly, but she would not be able to pass her 20s because her physical age is already something like 80s or 90s. That's what this progeria is defined as a premature aging syndrome. And this is a genetic disease. Okay? So, why this unfortunate illness is happening? Because of this disruption of chromatin territories. How did it happen? Because of, what is this? This is nuclear membrane. Nuclear envelope, should I say more correctly. So we do have a specific dye only that can stain the nuclear envelope, so that we can identify the location and the shape of nuclear envelope. And so this is so-called average people's nuclear envelope shape. And this is, guess what? Progeria patient nuclear envelope. It's all crooked, disrupted. The reason why this nuclear envelope structures got folded, collapsed, is because of one cytoskeleton protein, actually. I wouldn't even name particularly. One specific cytoskeleton protein underlying the beneath of this top of nuclear envelope, due to the mutation, genetic disease, that protein is missing in this patient. So we don't have any she or this patient do not have any supporting the cytoskeleton protein. So like our the cell collapse, this nuclear envelopes collapsed. But more important message is, okay, so what? This thing whole collapsed. But do we have to die because of this? It's not fair. It's just only a nuclear envelope. Why does everyone have to maintain this perfect round-shaped nuclear envelope? Can you not somehow live through this imperfect shape of nuclear envelope? What's wrong with this? Because if you do that, then this is another. Like for example, on this average, the reason why I keep calling this average is I don't want to call this as normal. As I mentioned earlier, once again, everyone is not normal. So yeah, we are only lucky that we don't have this particular patient, but that doesn't warrant us to name us normal. Anyway, this particular gene located in this area, due to this collapse of this nuclear envelope, now it has shifted into this different position. So this is only one small indicative features, but by using this the observation, we can learn that we can sense that whole entire the chromatin territory things like destroyed. Okay? We can identify this, and what happens is then now the result of such thing is disruption of gene expression program. Gene expression, if you're not really kind of into, I don't really know what exactly gene expression is. Okay, just leave it that way until you can become more familiar with this concept. But gene expression means each genes. What do you mean by gene is expressed? Gene is making RNA, and that RNA is now being used to make protein. So once again, gene expression equals to protein production. And we do have, as in a human cell, we do have about 20, 30, 23,000 different genes exist. So all these different genes have some specific program. So when to be expressed and when to be shut down type of such fine controlled program is disrupted by this structural collapse of nuclear envelope due to the disruption of interference of this chromatin territory. And not only in this occasion, but these are the example of another cases like cancer and some serious immune defect. Once again, the same such chromatin territory disruption are also observed. So in other words, this chromatin territory maintenance turns out to be very critical in like maintaining healthy cellular life. Otherwise, if you somehow if you fail to maintain such chromatin territory, like very unfortunate diseases like progeria, cancer, and immune disease happen, is the message. So already 19 minutes, sorry. So let's take 25 minutes break from now on.